All right, let's get started, everyone. Listening up, thanks. All right, we'll start with our weekly check-in. So nice little chance for us to have a little cuddle and chit-chat. So we have, first off, our random question. Where did my clicker go? So I just want to walk around the room. There it is. And pick some names out of my jar. So, question for today is favorite cuisine. So what is your kind of favorite kind of country's food? I guess is the easiest way to describe cuisine. So it's Mexican or Japanese food. The person answering that question today is going to be Lisa. What's your favorite cuisine?
Is there such thing as Indian?
Yeah, Indian, like curries and stuff. Yeah, Indian food, very cool. That's yum, I love good curry. Right, next person going to share that because I love that with a random question. Fua. Chinese food. Yeah, it seems to be like a common answer, like the Asian foods as like people's like favorite. I think it's just nice and easy to get. Anybody else want to share? Favorite food? I reckon mine's probably Malaysian, but I think it's because that's what I grew up with. So that's like the flavors that remind me of home. I also know a lot of kids like sushi. That was pretty popular.
Oh, I love going to Vietnamese restaurants.
Yeah, some people were saying Japanese is their favorite. But anyways, it's quite nice to get those like balls. So those soups are like real. All right, random question then. I'm sorry, not a random question. My goal for this week, person answering that is going to be Hannah. Both of us, yeah. Hannah.
It's have a good pink shirt day on Friday.
Oh, very nice. I like that. Hopefully everyone's got their pink shirts sorted out and your gold coin donation. All right, cool. Anything else that anyone wants to share before we move on?
Can I message?
Yeah, go for it.
Okay, so guys, if you didn't know, this week is Kindness Week. So us of the Pathway Committee have organized something for every single day of this week. And you know, obviously Friday's Picture Day, but tomorrow is something new that we're trying out called the Anonymous Friend, where we have these praise postcards that you can write a nice message to someone anonymously, just to brighten their day. So say I wanted to write something to Angela and say that she's been doing really great in chemistry, but I won't write my name on it, and then it will be sent to her on Friday so she can just brighten up her day and she knows someone's thinking about her. Or you can write it to a teacher, maybe you want to say something nice to Miss Garlic, but you don't want to tell her in front of everyone. Or Miss Garlic, you can write something to a student or a teacher that you potentially think that will deliver them for you.
That's very lovely that you guys are doing that. Should be a nice week. Did anyone get hot chocolate this morning? No. Oh. Maybe next time. Anyway. Cool. Let's talk about our learning journey. So we're switching gears. So to recap last week, what we looked at was the hydrolysis of esters. We only had one content lesson. If you guys remember the second lesson I gave you as a tutorial time. So today we are basically doing the last lesson that we have for organic chemistry. Then once we do this lesson, there is nothing else I need to tell you about organic. You're ready to hit the ground running and go. So this lesson is all going to be on the polyamides. So, two words that you have seen before, poly, which is that repeated unit, amide, which is one of our functional groups. And so basically we're going to see a repeated function group with the amide component. You guys have already seen this before in biology, so it'll be a nice little link between the two sciences. So do keep that in mind. As another reminder before we move on, next week, Monday, is when I'm going to give you guys your organic chemistry test or practice test. It's going to be a full organic chemistry exam so you guys can see what the scope of one of those exam papers are like. Remember, I am allowing you guys to bring a cheat sheet in for your practice test. So it's an A4 size piece of paper, single-sided. If you write on both sides, you're going to have the Sophie's choice which side you want to use then. And then I'll scribble out the other one. So do keep that in mind. Do start making them. Since this is our last content lesson, my plan is for the next lesson to give you, again, as a study period, so you guys can have a chance to make them if you don't want to make them outside of class time. Yeah.
Some of us will be on Bandcamp on that Monday.
Really?
Yeah, because we're going this Sunday, Monday, Tuesday.
Okay, then you can just make it up on your back. Okay, yeah, that's not a big deal. All right, cool. Anything else? All right, just get ready for that. Today is our last lesson. And before we move on, let's think about our weekly Takatoki and think about what we're kind of living by for this week, this piece of advice. So we have Naki ana mua te toto mai ana muri, which is first clear the weeds, then plant. Another way to think about that is make time for mind weeding. And I think that's such a great piece of advice. There's no point in just starting your garden without first taking a survey of what's already there. Because if you're starting to plant things and there's weeds there, those weeds are going to overgrow and you're not actually going to have a really productive farm or garden. So do make sure you're clearing out those weeds. Make sure you're thinking about your mind as well, getting rid of all that stuff that might be bothering you. So that way you can kind of sit and get some productive time going. I know for me, I can't think when it's messy. So I always tidy before I start doing my work because clutter around me is clutter in my mind. So do keep that in mind. All right, lesson plan for today. It looks a little bit packed, but actually these are little small chunks of information that I'm going to go through. So don't worry too much about it. We should hopefully be able to get it all done today. I think we should. Because most of this will actually be refresher because we've seen most everything. All right. Cool. And then on our success criteria, we're looking at number 7, which is the polymerization, but in this case, we're looking at 7b, which is looking at the amides. In particular, we're going to be looking at proteins. What I want to point out with those proteins, when we start looking at these polyamides, you do not need to name the amino acids. I'm going to show you guys all the amino acids. They're going to mention the names of the amino acids on the exam, but in no means do you need to memorize them and regurgitate that information. It will always be given to you. All right. So here's my joke for the day. What do you call an acid with an attitude? Amino acid? Yeah, I'm sorry. All right. All right. So, things to remember about amino acids. Amino acids, you guys have seen in biology, they're the building blocks to proteins. Now, with the amino acid, it is exactly what you'd expect it to be naming-wise. You have the amine group, which is your your amino group, which is your nitrogen and those hydrogens over there, and you have the acid component, which is your carboxylic acid. So it's again one of those molecules that you see that there's 2 functional groups present. And we know when we were talking about the polyesters, they needed to have 2 functional groups to be able to make the polymer. The same idea. These are the ones in the human body. I believe there's 20. Yeah, there's 20 in the human body. We make most of them. The ones that we don't make, we need to get through our diet. But what you can see that's unique about these amino acids is they all have that nitrogen group, so the amino group. They all have the acid component, which is that purpley color. And then what's being, let me back to those 2 things. The third thing that they all have is the hydrogen. And the last thing is the different components. So what's making it the unique amino acid? So that is basically the kind of component that's going to give it those properties. So you can see here in red, those are showing the different amino acid kind of structures that you have with each one. And they are named. Again, this is what I'm referring to as you do not need to memorize the naming of these. They will always give this to you on the assessment. The other thing is, what do you guys notice with all of these amino acids? Here. They're just going straight down. They're oriented in the same way.
Is there a side chain?
They all have the same basic component. What do you see in their naming?
The L.
They all start with L. Where have you guys seen that before?
With the N. With
the optical isomers. Yeah, so remember with the optical isomers, the naming, there was like the L, and I can't remember the other letter they used for it. But do you see how all of these, except the one, are going to be classified as optical isomers? Because they have 4 different components on that central carbon. Yeah? So the reason why I want to point that out to you is that they often use amino acids for the optical isomer question on the exam. So that's why we're going to do it as practice anyway. It's really interesting to note that humans use the L version of the isomer. We don't use the other one. Different species use different ones, some use both. It's just an interesting thing to kind of note about biology. Cool. All right, another refresher then is how do we figure out what amino acid to grab. So this is some biology stuff here. We look at the DNA, and the DNA will become messenger RNA, and that messenger RNA tells the ribosome what amino acids to put in what sequence. And so we have the 3 codon sequence, and that tells you what amino acid to grab. So that's just kind of to show you guys how this is kind of looking together with your biology. Cool. All right, and then this is what we're trying to make at the end, is this nice, pretty kind of 3D structure. And it's really important that this is the right 3D structure because everything fits in like a lock and key. So we see here how we have some other molecules, and those other molecules can fit in perfectly because there's the right shape for that protein. And the right shape of that protein is influenced by the amino acid chain. Are we good so far? All right, cool. Yes, I've talked already about the optical isomers. So here's an example, D, that's the other one. So there's L and the D isomer, and it's just a matter of which one's going clockwise, which one's going anti-clockwise. We are using the L isomer. Okay, Hannah, you look kind of...
I don't know, it's just they never told us about the L and D's and the isomers.
So they probably wouldn't bother mentioning in biology because then they would have to teach you about optical isomers.
Yeah.
And that's a chemistry-based question.
Oh, okay.
So like,
What does corn stand for?
I don't know. I've never seen corn. Or I've seen corn. I'm not sure what corn is. It's probably an acronym for something. Think about the rotation.
It's an acronym. I don't know.
It must be an acronym for like regarding rotation.
Okay.
Would be my guess. Or maybe does it have to do with carbon? No, I don't know. You can Google it later and find out. All right. So if I'm thinking about these amino acids, which one is the one that doesn't form optical isomers? The glycine. Because we see how glycine. Why is that, Hannah? How do you know?
Because it doesn't have the L.
It doesn't have the L in front, and what's the...
Yeah, the 2 H's.
Yeah, there's 2 H's because they both have the hydrogen there. So, fun fact, I point that one out because, again, it's a common question on the exam. So I think, oh no, maybe next slide I'll talk about it. All right. The other thing to remember with amino acids is that they have both the acid and the base component to it. And that's then going to influence its properties as an acid or as a base. So if I put my amino acid into a, the amino acid can lose one of its hydrogens, acting as an acid, sorry, not amino acid, the acidic component. The amine component can also be a proton acceptor and gain a proton. So in both cases, we can add or lose hydrogens. So like I said, it has both the components of an acid and a base. What this then means is that if I put it into a water, if I put my amino acid into water and have it at pH 7, both acid and base components will be affected. And so I have my carboxylic acid losing a hydrogen and the amino group gaining a hydrogen. We call these things Zwitter ions because they're able to have both a positive and a negative charge. It makes it really, really soluble in water, which is great news for us because we need them to build proteins. And they also have really high melting points, which again is really good news for us because we don't want our proteins to denature very easily. We want them to hold their structure. There's actually a lot of science whenever you're working with proteins and things like that, where they'll adjust the pH in the solution to see how the protein behaves and also to help separate different types of proteins. Because depending on what amino acids are there, that's going to influence its overall charge. So it's cool science stuff. All right, cool. I will give you guys a little break from me talking, and I want to show you guys an example of amino acid questions on the exam. This should be in your little booklet, just FYI. This is taken from the 2016 exam. It's question 1C. And what you can see here is they've given you 3 different amino acids, and they want you to draw an enantiomer for the serine amino acid, which is this one up here. The other part of the question is which one of these does not display optical isomers, which we talked about in class, so hopefully you can identify it based off of that list and the why. So take a moment do a little bit of a refresher, draw some optical isomers for me. And if you forget, don't worry, I'll go over the answer in a moment. All right, let me go over the answer to this. Which is okay. It happens. All right, cool. Answer time for our optical isomers. So be mindful, I only need you guys to draw serine. You don't need to draw the alanine, which some of you guys did because you're like, I need to draw two things. And you just drew the other amino acid, and that's not what it was asked for. All right, so I personally just take whatever they have written down, and I do a bold and a dashed line to make my life easy so I don't have to flip anybody around. So you should have carbon, NH2, we'll do this one dashed, COOH, we'll do this one bolded, and that one's H, and we'll do that one within plain, CH2, OH. So that's one optical isomer, and then all you need to do is flip it around like it's seeing itself in a mirror. So I'll draw that on the other side. So the nitrogen group and the hydrogen can stay the same. The only thing we're flipping around is the stuff that's in the horizontal plane. So I'm doing dashed lines because we're pulling everything with it. And that's going to be COOH. I'm just doing it reverse so that way we can see it the other way around. And then pull, again, the whole thing for the alcohol. And those are my two optical isomers. Sorry, I've drawn them on the opposite page, or sorry, opposite sides of the paper. It was the only way I could get it to fit. All right. Are we okay with that so far? Very common question. Don't be surprised if they tie that with your amino acids, because amino acids obviously lend themselves really well to optical isomers. All right, in this case, the answer is glycine. We talked about it in the notes. It has to do with the fact that glycine is not a chiral carbon. It doesn't have a chiral carbon. So that's just something to be a nice little refresher. Make sure you are specific and you've answered the question. So glycine is not a chiral carbon because it doesn't have 4 different groups attached to it. And in this case, it's 2 hydrogens. So be detailed with it just in case. I see this generally only as a merit level question, just FYI if you guys are a little bit curious to know how the points break down with that. But it's a really easy merit if you can just draw 2 things like that and explain something. All right, moving on. Anything you need me to follow up on that? Pretty straightforward. Nice refresher on optical isomers. You'll definitely get a question on that. Polyamides, also known as polypeptides. Can I get people with headphones out and listening? I know some of you guys are watching other videos of lesson material and doing work, but we got new material to cover. Lisa, shut your headphones out. Good. Hello. Hello. She can't hear me. Alright, cool. So, polyamides, what we're looking at, also known as polypeptides. This is how you guys are probably used to it for biology. What we are doing here is we are attaching 2 amino acids together to form a peptide link, also known as an amide link. You'll see a lot of interchangeable terms, making a polypeptide, and we just repeat that over and over. The R group that you guys see is just representative of the side chain, so it's just to give you guys a basic foundation of what these polypeptides look like. And you'll notice when this happens that we remove water as our byproduct. You can call that a dehydration reaction, you can call it a condensation reaction because water is the byproduct. If I want to break up my polyamide, we use water for a hydrolysis reaction. We go in and we break it apart, just like you guys saw with the esters. These things are really great. They're strong, they're elastic, they're really tough, and they're resistant to abrasion. They obviously are our building blocks for all the various bits of protein. Are we okay with that so far? All right. The example that you might have come across is nylon. I'm actually wearing some right now with just the stockings. Again, just polyamides, which are really, really useful. All right. Let me show you guys this exam question, but with this exam question, I will do it because I want you guys to know how to do this. So, here we go, 2016 exam, and once again, we are taking the amino acids they have given us, and they want us to draw two possible dipeptides, and I heard some people talking about it, and they're like, how am I supposed to draw two? Let me show you how. It's actually ridiculous, easy. So, they have given me glycine, alanine, and serine. We see this from the previous question that we asked on that. In this case, they want us to draw two possible peptides, and they want us to grab the glycine and the alanine. So we're ignoring serine in this example. Now, what I like to keep in mind to make my life easy, and you can answer this multiple ways, but the way I like to think about it is, what's the general setup and formula? So my general setup, when I think about a polypeptide or polyamide, is that I need to have a nitrogen. Hold on, I'm trying to think now. I messed this up already. Where's my twink at? Yeah, if you want, turn the light off. Because I know it's a little bit bright. And I can also turn this down if that helps a little bit.
Nice.
Is that better?
No. Marginally.
Marginally. All right. So, whenever we think about an amide, remember you have a C double bond O. We have a nitrogen connected to it. And then we have the carbonyl, like the main, sorry, not carbonyl, the main carbon group. And then it would have a little nitrogen here. Are we okay with that so far? So all I've done is I've drawn two carbons. I haven't actually even pulled amino acids. When I look at this guy, I should know that both of these hydrogens are going, or sorry, both of these carbons will have a hydrogen attached, because we know all amino acids have that. Since this here has the amino component, then I'm going to end with a carboxylic acid on that side. And then on this side, when I look at it, it's ended with a carboxylic acid, so this end is going to be my amine. Are you okay with how I've done that so far? So that there is the general setup, and then the only thing I need to change is what the R group is coming off of it. So in this case, we're doing glycine and alanine. So I'll do glycine first, and that's just got the hydrogen. And then for alanine, it has the CH3. Are we okay with that so far? All right. Once again, I'm going to draw the general structure of what I would expect to see in a dipeptide. So I should have, I always like to personally start with the amide component, because it's then easy for me to set everybody up. So I'll just highlight that. So this here is my amide, and they often ask you to highlight the amide. Then I just draw the rest of the various components to set it up. So the carboxylic acid, in this case, the amino group in that case. So that's just a very generic, this is setting up for a dipeptide. The only thing that you do differently then, when you say the two possible dipeptides, is I swap around the R groups. So instead of starting with the hydrogen, I start with the CH3. And in this case, my second amino acid is the glycine. So all you're doing with this dipeptide, in this first one you have glycine, then alanine, and in this case we're doing alanine and then glycine. So it's just about the order that you put it in, and these are two different dipeptides, because the order of the amino acids matter. Because the order is going to influence then what type of chain we get and how it's going to influence the folding of the protein. So basically all you're trying to do is swap around the R groups. So you do one and then swap the R groups around. Does that make sense? I don't know if that was like a more complicated way, or if it was easier for me just to draw each amino acid first and then put them together. But this is the general setup that you're looking for. And I see a lot of mistakes happen with that general setup. So don't like overthink it, and don't try to do some weird additions like putting two amino groups together, two carboxylic acid groups together. It should go, you know, amino, carboxylic acid, amino, carboxylic acid. You can do it the other orientation and start with the carboxylic acid and end with the amine. That's fine as well. But the thing is, these are two different dipeptides, because in this first example, the glycine has the amino group ready to add to it. In the second example, the glycine has the carboxylic acid group ready to join to it. So that's why these are two different dipeptides. And that's all you got to do for that question. This one often, like I said, goes up to a merit level. So some of them are real easy merit points, and this is another example of an easy merit point. All right, what questions do you guys have after seeing that? So if you go to No Brain Too Small, they have collated them, and so you can do the collated polypeptides, and you'll see it. So this is categorized with the other polypeptides. Oh, sorry, not polypeptides, polymers. All right, can I move on? I know I want to just get all the notes done so you guys can sit and get some work done. All right, and I just want to show you guys some really cool things too. It's really important because that influences how that protein is going to fold. If I have two amino acids joined together, we call it a dipeptide, di meaning two. Anything more than two, we start calling it just a polypeptide, because now we're starting to deal with the chain. This is what you're thinking about when we look at these polypeptides. So we have a chain of amino acids, and in this case, we have some that are orange and some that are blue. So some of those amino acids are hydrophilic and they love water, and some of those amino acids are hydrophobic and they hate water. Looking at that image, do you think it's the orange or the blue that hate water? Not sure. How many say orange? How many say blue? 50. It is the blue. So if you look at the way this amino acid is structured, the blue is hidden on the inside, and so that's the hydrophobic component to it. Hydrophilic will be on the outside. The other thing to think about when these amino acids fold into the protein is that there's some common patterns that we constantly see, which is the the beta sheets and the alpha helix, which we can see here. Some helixes, and we can see some folding, zigzag, accordion style. So that gives us a tertiary structure, and then that structure then can join with other proteins, and make a nice big quaternary structure to then do functions and things in our body. Cool. So lots of different examples of what we can use this for. Obviously, with these proteins, we use them for hormones and messaging around our body. They're also used to build structure. They're also used in chemical reactions. They are super, super important to the body. For example, you guys would have done this in level 2 biology. If something goes wrong with the DNA sequence, it can change that amino acid, which can affect its folding of a protein and make that protein defective. Sickle cell anemia is an example of that. This is a point mutation that has changed one nucleotide. It's told it to grab a different amino acid, and when we looked at that full chart of amino acids, some of them are hydrophilic, some of them are hydrophobic, some of them can do sulfur kind of dye bridges or whatever they're called. So it can influence the shape quite dramatically. Cool. All right, we're going to take a little break, and I have a video to show you. So let me introduce a little bit to this. This video is from a software called Folded. Now, fun fact, we have no idea how the body knows how to fold proteins. Like, no idea. We know the DNA sequence, we can figure out the amino acid sequence, and then from there we can kind of guess some things that we think will happen, but we don't actually have a way to translate from DNA to protein, which is really neat. And in fact, the group of scientists, because scientists work in teams, that figure this out, it's going to be like figuring out the structure of DNA. It's going to be groundbreaking for us. It means that we can reverse engineer things, which would be cool. But whoever figures that out is going to win themselves a Nobel Prize, because it's going to be such a huge big thing. One of the ways we're trying to figure out protein folding is through computer programs and computer generation to predict models. And so what this group did is they took the information about proteins, they took the information about programming, and they turned it into a game. And so they're using the human mind to see if we can predict patterns to then create the shape of the protein. So I'll show you guys this video, and I'll also show you guys the website. They collect our data of us playing this game to try to see if there's an algorithm or pattern to how proteins are folding. So I'll just show you guys this because it's cool. All right, cool. So we'll take a little break from me talking, and I have some polymerization revision. There's 3 questions on there. It is in your booklets as well if you're having a hard time seeing it on the board. But it's a nice little reminder of how do we make our polymers, and it has both the making and the breaking, and it has both esters and the amides. So I'll give you guys a couple minutes to get that done, and then I'll talk about the last component of this lesson, which is the hydrolysis of the polyamides. All right, so just is about the hydrolysis in like crazy amount of details. But the main thing I want to focus on is that you understand how hydrolysis works with the polyamides. It's more or less similar to what you see with the polyesters, but there is that key difference between the acid and the base that we need to pay attention to. More so important because you have a functional group that's the amino group, and you have the functional group that is the acid group as well. So that's why we need to be a little bit more careful with how we hydrolyze it. So again, I'll teach this to you guys looking at an exam question so you guys know what to do. Hydrolysis 1 is, please don't freeze. Okay, we don't need the sound. Okay, we're gonna have to restart that. I'll try to be quick because I know you guys only got a couple minutes left. Okay. So I've taken this exam question from the 2021, so it's quite recent. And you guys should have this also in your little booklet of notes. So very similar to the polyester question that you have, but in this case, instead of having an ester link, you have a amide link, which I'm just going to circle right here. Same process that you're going to do as you do previously with esters is you're going to look for the carbonyl, and you're breaking between the carbon and the nitrogen. When you guys have the polyesters, you were breaking between the carbon, that's the carbonyl, with a single bonded oxygen. So, but you're looking for the same thing. When you break it apart, be mindful that they often give you 2 different amino acids. So you're going to have to draw both of them. So I can see here that this one has this as their R group, and they have this one as their R group. So that's the first one, that's the second R group. So 2 different amino acids that are present. Now, it has the same conditions of what is a hydrolysis reaction, what reagents do you need, etc., etc. And then they ask you to draw what the product would be in acidic conditions and in basic conditions. So what I've done is I've gotten another sheet of paper for us to have an example of what to do. In both cases, you're going to draw the amino acid, but you're going to neutralize one of the functional group components. So I'm just going to draw this first amino acid, which has CH3, NH2, C double bond O, OH, and I'll draw the second amino acid down as well, and then I'll show you what to do with the acid hydrolysis. So this one had CH2OH, that one had NH2 and COOH. Are we good with that so far and how I split it up? And in fact, you can do the exact same thing for the base hydrolysis, just to set it up. Draw both amino acids so you know what you're doing, and then fix it to show the neutralization. So I have broken up my amino acids. Oh, I should actually draw the C double bond O because I think it's useful to see it that way. Cool. So this is just me setting up what I'm doing. Now, if I have something that's in a acidic hydrolysis, what that tells me is you're going to be neutralizing the base. So in this case, the basic component is the NH2. So when I neutralize a base, what we're going to do, remember, it's going to be a proton acceptor. So it's going to accept the extra protons in solution. So NH2 becomes NH3 in both of these examples, and you're going to put a little positive sign on there to show that it has an extra hydrogen and it's been neutralized. Do I do anything to the carboxylic acid in an acid solution? No, it gets left alone. All right, in a base solution, we are neutralizing the acidic component. Which in this case is the carboxylic acid. So with that carboxylic acid, remember, acids are proton donors, so you're going to lose a hydrogen. So this guy's going to lose the hydrogen, that guy's going to lose the hydrogen because it's going to donate it to solution. You show a little minus sign because that is it becoming a salt. Depending on the detail they give you, they might tell you what base you have, and so you might show it, like for example with the sodium, if you have sodium hydroxide. And then we leave the amino part alone. So that just stays as NH2. Make sure you do it to both of them. Make sure you leave the R groups alone. The only exception when you shouldn't be, or when you will not leave the R groups alone is if the R group has a carboxylic acid or an amine in it, which is very unlikely for them to throw you those two curveballs. All right, what questions do you have after seeing that? None? It's a little bit different from the esters, because remember when you do the acid-base hydrolysis in the esters, the alcohol component stays an alcohol component in either situation, and the only one you got to worry about is the carboxylic acid component. In an amide hydrolysis, you need to worry about both the amine and the carboxylic acid because they have both acidic and basic properties. Does that make sense? We'll do this. You guys are going to have a study lesson in the next period, so you can make your cheat sheets. You can also have a chance to review some of this sort of stuff as well. And I will scan everything and put it online. I am recording these lessons still, so if you need to watch anything again, it's there. All right, I'll just summarize the lesson, and then you guys can pack up. So this is the Mahi choice for you guys, just to know the different levels of the work so you know what to follow up on. Please comment when you get stuff done and show me it so I can give you credit and stickers. But to summarize today's lesson, you should be able to do number 7 off the checklist, and in fact, you should be able to do the whole checklist now, because we are done. That is everything I have to tell you with organic chemistry. Congratulations, now you can study. All right. Make sure you wipe down the tables and spray them. Don't put the chairs up yet until you've sprayed the tables down. So spray the tables. Yep. Spray and wipe the tables and then put up the chairs. Windows as well. Just be very careful. I fell off the bench the other day. It wasn't fun.